So before introducing next speaker, I want to thanks the Food Smartphone project to give us the opportunity to make this workshop, and of course the European community, because this project is an ITN from the European community. Okay. So next speaker is Pieter Moons um, from Belgium. He obtained his PhD in bioscience engineering from the University of Leuven in 2009. And after that, he made a postdoc also in this university in lines such as genetics and microbiology. So after all, Peter joined Sanico NV, that is a pharmaceutical company. It was in 2010 where he worked as a scientist in the QC department. In 2011, he joined the International Lab of Medical Microbiology at the University of Antwerp, where he managed and was scientifically involved in several academia and industry consortia focused on the development of rapid diagnosis for infectious diseases and participated in a clinical trial network aimed at combating antimicrobial resistance. Although he is still part-time affiliated to this university with several research lines focused on infectious diseases diagnostics. In 2018, he was appointed the manager of Biobank of Antwerp, which was established at that time in response to the new Belgian biobank legislation, and he collaborates between the University of Antwerp and the university and the hospital from Antwerp also. There, he and his team currently face the challenge of integrating the existing collections from both institutions, the university and the hospital, aiming to create a high-quality biobank that serves the needs of both academia and industry. So, welcome. And
thank you, Roger. And also thanks for the introduction. So I think I can summarize that I've lived both sides of the world. I'm still part of the research. I'm still working in academia, 20% as a research in the University of Antwerp. But now I'm also on the other side, so as a manager of the Biobank Antwerp. And I'm also a member of the ethics committee in Antwerp. So I see a lot of projects that make use of human material or make use of humans. And that's why I'm invited here to give this presentation. Because after the talk of Louis, who talked about the use of animals, we're still actually using humans, a lot of humans actually, in research. And of course, this is very well regulated, and this is also what I'm going to talk about. Good, thanks. So briefly, I'm not going to give you a complete picture of the use of human material today. I can only give you some concepts related to this use of human material for research. So I'm give you parts of the puzzle and give you also some pointers or give you some directions to where you can find more information. And I said I see a lot of young researchers that come to me both as manager of the Biobank or as member of the ethics committee, and they say, okay, I'm going to do this marvelous research, I'm going to solve this great medical problem, and I need human material. And then I'm like, okay, and what exactly are you going to do? And then they're like, yeah, I don't know exactly yet. And then we have to take into account that, of course, all this human material is very valuable. People are often very, very sick, have even died sometimes, and they provide this material. So this material is valuable. We have to recognize that. So the first thing I'm always asked them, really work on your research question. You have to know very well upfront before you start what you're going to do with this material, because often you cannot go back in time and rectify things. Once you started your certain research trajectory, you cannot shift that easily. If you failed to take a certain sample, patients are gone, and you might not be able to answer that specific part of the research question. So to me, this is an important message to young researchers. Human body material. We're actually doing a lot of tests still on live humans, clinical trials. So people are usually not, well, they're aware that we're doing clinical trials, but indeed we're still experimenting on live humans. But we're also using in our experiments a lot of human body material, and that can be various things. So it can really be tissues. So in this case, this lung from a smoker, probably because it's lung cancer. So that's one type of material. We can have stool samples, urine, blood. So these are things that are excreted by the body. So it doesn't necessarily need to be part of the body itself. But basically, what you need to think about as a researcher is, what are you going to do with the samples? Sometimes, for example, you need both a tumor tissue and you want to correlate that, for example, with the gut microbiome. So then you might also need the stool sample. But then again, your next question, research question might be, okay, I want to have these biomarkers. So you need perhaps blood or urine, depending, because these are easier samples to perform your research on. And basically, you're not interested in the samples themselves, but you're looking into these molecules. Might be proteins, might be DNA, might be small molecules. Those are the things you're actually looking for, and those are also the things that you should take into account when you're establishing your research question. Because then I'm coming to the next thing that I often ask these young researchers: what do you need? And they say, I need blood. And then I'm like, yeah, okay. What type of blood do you need? What do you want to do with the blood? And that comes back to these molecules. Because what you see here is actually quite a number of tubes where you can collect your blood in. And depending on the type of tube that you use, that really decides the type of research that you can do. Because, for example, you might be interested in collecting plasma. So that's these green tubes here. But if you use heparin, which binds this antithrombin, that is known to inhibit PCR reactions. So if you collect blood in heparin tubes, you can forget about all these DNA analysis. The same lithium, EDTA, these are known to impact on cell viability. So if you want to do some experiments using live cells, then these tubes are out of the question. So these are all things that you have to consider when you select your human material. And this can really ruin research. And then the next question is, to these young researchers, what are you going to do? Are you actually going to do this experiment? Because all young researchers think they need lots of patients. They come to me and they say, I need 500 cases and 500 controls. And then I'm like, okay, so you're going to do this primary study. You want to collect these samples, so you need 500 or 500 cases and 500 controls, so 1,000 patients. So that takes actually quite a lot of time and resources. So who's going to collect, who's going to address those patients, who's going to collect those samples? And in a lot of cases, these students didn't think about it. It's just like, okay, somebody in the hospital will do that. And who is going to fund that? Yeah, probably my PI is going to fund that. But the advantage, of course, is you have full control. You can really decide, given the discussion, the fit for purpose discussion, you have full control about the sample preparation, the pre-processing. You know which tubes can be used. You can decide on that. You can decide, okay, you want to centrifuge that at 4 degrees within 10 minutes of obtaining the sample, because then you preserve your biomarker of interest. So of course that's a big asset of doing this primary collection. But in a lot of cases, it actually comes down to the PhD student activating doctors in the hospital. So doctors, they're all enthusiastic when you address them. It's like, okay, it's great research, I want to participate. Yes, I have 500 patients a year with that specific disease. But when it comes to gathering samples, when it comes to collecting patients, to provide them information, they usually are happy to do that in the first 2 weeks, and then it gradually diminishes. When we're talking primary use, I included this perseverance. So you might spend the first 3 years of your PhD just chasing patients, trying to get those samples in, before you can actually start doing any analysis that you want to do. The alternative is secondary use. You're actually building your research on what somebody else already collected. The advantage is it's often faster and cheaper. Somebody spent the money, spent the time, did all the hard work. Of course, the problem is you didn't have control, or you don't have control. You have to work with the sample that is there. And I'm going to just go through a couple of these sources of human material, just for your reference, so that you know where you can find those, and also to give you some pointers related to how easy it is to get those samples and their value. So you have the diagnostic labs, you have the tissue banks, biobanks, and clinical trials. So all of these can be really sources of human material. Let's start with the diagnostic lab. So this is really the clinical biology lab, the pathology lab. So basically, a sick patient comes to the hospital, a sample is taken for diagnosis, and part of that sample usually remains. So it's in that lab, can be a blood sample, and it usually needs to be available for the diagnosis. Might be that a follow-up diagnosis is needed. In clinical biology lab, that's usually days, so they store the sample for a day or 3 before they discard it. In pathology labs, that's, well, at least in Belgium, the law says it's 30 years that the sample needs to be kept. Of course, you can chip off small pieces of that sample for research purposes if the pathologist agrees to do so. But of course, you don't have any impact on the sample processing conditions. They really follow their own procedures, and you can only hope to be able to use some of these samples. What we often see still is that these samples are available and students know, or the PI knows the doctor in the hospital, and they just make an arrangement and they say, okay, I need some samples. PhD student goes to the hospital, takes some of these samples away, takes them to the lab, and does the research. It's actually, of course, no longer acceptable, but we still see that practice actually going on a lot throughout hospitals throughout Europe. A different source of human material is actually therapeutics. So basically, these are the tissue banks, and there is a very nice legal framework. I'm not going to go into the details of the legal framework, but I listed it so you can have a look. But basically, the legislation is very strict because these materials normally are intended to go back into humans. So this is really therapeutic use. So you can imagine that those samples are being collected with high precision according to very specific procedures where you have no say whatsoever. There's some testing going on, there's quality assurance going on. Also the storage of these things is according to very strict quality regulations. But then again, you really have no say in that process whatsoever. And of course that primary use is very important because that saves lives. So your research or whatever goal you might think you have for that sample is of secondary importance. So often these samples are not readily available or are only available when the quality diminishes. So that's something that you have to take into account. And then I added this. So I don't know how it's in Spain. In Belgium, we're all donors. I'm not sure that's the case in Spain. No. So whatever reason, but donation is crucial and really saves lives. So that's why I added this. Another source of human material, clinical trials. Very, very strictly regulated throughout Europe, European legislation that covers these clinical trials. At the moment, that's still the directive of 2001, and it should have been replaced by the clinical trials regulation since 2014, but there's a slight delay because of the clinical trials information system, which is normally due end 2019, which is nearing. And the current status, I've checked, I think 2 days ago, on the website. The current status is that it's indeed should be very near, and they validated some aspects. So this should really change the clinical trial regulation in Europe, should make it more harmonized. So it's basically pharma lobbying, we have to be honest here, but should make it easier for pharma to run trials in various countries throughout Europe, which probably is an asset because, well, we all know Europe is one, but also divided. The problem is, of course, a lot of these trials are driven by pharma, and pharma basically decides what happens to the samples, and usually you never see those samples again, especially not as an academic researcher. And they do very solid research on these samples themselves, so I'm not saying anything, I don't have anything against pharma. But usually those samples are not available to young PhD students unless you have these academia industry trials, like for example via the Innovative Medicine Initiative. And I've actually participated in some of these IMI funded trials, and they're definitely worth joining. They first of all bring decent money also for academic research, but you really have a say also into the research, so worth looking into. And then last but not least, the biobanks. There is no European legislation related to biobanks, so that's a big drawback. That means that basically every country has its own legislation, if they already have legislation. And that basically also means that in a lot of cases, it's just researchers that establish collections. There's no such thing as a national biobank or some national biobanks. Well, they're often called biobanks, but in most cases they're just collections of samples owned by investigators or groups of investigators. The advantage is there's very large collections available, very valuable collections available, often also with multiple samples. Might be case control, might be consecutive samples of the same patient coupled to specific disease, often multiple samples of the same patients of various areas of the body, so you can really link, for example, or search for certain markers within multiple of these samples and then link those together. A drawback is often that these samples are owned by a specific principal investigator, and that is often not easy to get those samples out of these collections, because that principal investigator always has some idea of what he wants to do with the samples. You really want to aim for collaborations here. Usually it's easier to get those samples out of the collection if they also see some benefit, and that's of course since they invested a lot of time and money in getting these collections up and running. What we do have is BBMRI ERIC. So that's one of these European research infrastructures. So everybody knows CERN, but this is the research infrastructure for biobanks. So although there's no European legislation, there's this European biobank infrastructure that basically works in the same way as the collections of the animals, basically. So you have all these national nodes that together form this biobank infrastructure. And although they took off slowly, they now have established some very interesting resources. For example, this sample negotiator, so you can really go to their website and then say, okay, I need a specific type of sample. And then it automatically searches for where these samples are positioned within the national nodes. So it checks all of these countries. And a node is not necessarily one biobank. So for example, in Belgium, the Belgian node consists of, I think at the moment, 17 biobanks. So these represent actually a significant amount of potential resources. So it automatically reaches out to all of these nodes, identifies the samples that you require, and then you can, well, the sample locator does that, and then you can really reach out to these researchers as a group via the tool to try and obtain those samples. So in that regard, if they continue to grow, the aim is to really have this large European research infrastructure, which has a very, well, should have a very nice, massive collection of potential samples, very well annotated, so they're also looking to specific data that are coupled to these samples. So should be a very valuable resource for young research in the future. What they also have, if you need that, is support related to ethics and legal questions, especially also if, since the legislation differs quite a lot from country to country, so they can also help with those things. So, source of human material. The main question, I think, is here, fit for purpose. So all of these were collected with a different purpose in mind. So you really have to be careful that when you want to use those samples, that that these really fit or address or help you address your research questions. And if not, the only alternative might indeed be the prospective collections and basically lose the 2 or 3 years of your PhD. So that's a brief introduction of what the samples are, what is important related to the samples. But then, okay, you have come up with your brilliant research idea. You have identified the samples or the route to your samples that you want to use. And then you come to your next hurdle. Well, researchers like to call it a hurdle. It's the ethics committee. So this is a group of wise people that really look into your proposal. So you really have to submit a significant amount of documentation that helps this group of people, which consist of very specific profiles. So you have people with medical expertise, Often there's people with legal expertise. For example, well, I'm there now with biobank expertise in the ethics committee. So you have a number of these so-called expert people that really look into your file. And your file consists of your protocol, consists of your documentation, contains all information that you, for example, provide to the patient. And they investigate, well, they look into the scientific soundness of your proposal, so that's one thing. But mainly they check whether your file or your proposal is according to the ethical principles and according to national and international laws. And one of the first things, or one of the things they check very carefully is this informed consent form. And it's really standardized what type of information needs to be in that informed consent form. And one of the first things that need to be in there is the information that you provide to the patient. So you come in a hospital or you come in a primary care facility, you have a patient before you, and you start explaining the study, and you also give him some documentation that he can read carefully, and he's usually allowed to ask questions. So the aim is for the patient to really, or the volunteer to really understand what the research is all about. But that also means it needs to be written in the language of the patient. So if you have a trial in 12 countries, you need a very good translation of the document. And if you have children involved, they also, of course, don't understand the language that is used for adults. So you really need specific information forms for children. And what we see, especially for these young children, is that, for example, often pictures or pictograms are used to bring that information to these young children. So the first thing they need to understand is, of course, what are your intentions? What is the purpose of the investigation? Why are they participating in your research? And that's often something that is not that difficult to explain, although the language or the scientific language should be avoided in this case. Important is the treatment, absence of treatment, or so in some cases you cannot give the standard treatment, and that of course is a risk. So the ethics committee then needs to decide, okay, is the risk, or does the risk outweigh the advantages of, for example, an alternative treatment that you want to provide, or an alternative strategy that you want to provide to the patient. So risks also bring insurance, and can be very small risks. Even obtaining blood from a patient is a small risk. Usually it's only a bruise, but risks can be relatively significant for certain trials. And that's why the ethics committee will also look into insurance. Normally you need a no-fault insurance for all of these investigations. Of course, expectations. What do you expect from the patient? If you only need a sample of blood, that's easy. If they also need to fill like a questionnaire of 30 pages, that will take them 2 hours. They need to know upfront. So you really need to clearly list what you expect from the patient, but also what the patient will get out of it. And in most cases, there's no direct benefit to the patient. It's basically to progress the scientific field, and perhaps in, let's say, 20 years, there might be some solution for that specific rare disease that the patient is currently facing, but he will not personally benefit, or she, from that solution that you're making the first steps now. What can be another advantage, of course, if somebody has to spend 2 hours to fill a questionnaire, it is acceptable to compensate them for their time. What is not acceptable is that you buy the samples. So it's a thin line sometimes. So one thing that we once did ourselves in Antwerp, the patient, we required about 1 hour or 30 minutes of time from the patient, and we provided them with 1 or 2 lottery tickets. So they had a chance of, let's say, €1,000 or a couple of million euros. Whether they gained something out of that, of course, we don't know. But it's a small compensation for their time. We didn't really pay them for the samples because that's, of course, not accepted. Confidentiality of the data, and I'll come back to that because that's, of course, becoming more and more important. But also the right to evaluate the data. So if you capture data from the patient, you have to give them the opportunity to look into these data and to say, okay, this and this is correct, but this might not be correct, and you need to change this into something else. And then, of course, voluntarity of the participation and the right of withdrawal. So they're volunteering. You can only be happy that they participate, but they can step out at any moment without consequences to them. So it's not that the doctor can stop treating them. They can just withdraw. They don't even have to give you a good reason or any reason whatsoever for that. So that basically was informed consent. There is another option that exists. So informed consent is basically opt-in. You ask permission from the patient, and he or she agrees to participate in your study. The alternative is opt-out. It doesn't exist in all countries, and I'm not sure it exists in Spain. It does exist in Belgium. So basically, well, in Belgium it's related to organ donation mainly. So in Belgium, they're all organ donors. Same here? Okay, thanks. But if you're unwilling to donate organs, you have to go to a specific, well, either website or to the city hall and to register as not being willing to provide organs. And usually in Belgium, once you die, they still ask your family whether they can use your organs or not. They're actually not obliged, and they only don't do that in case you actively, because that's an alternative, you can also actively say, okay, I want to be an organ donor, and then they don't ask the family. What we do have in Belgium as well, and that's probably the same in other countries, so the presumed consent also works for the diagnostic samples. So basically, if you come in a Belgian hospital, you get these small leaflets that nobody reads, but that contains actually some information. And one of the things it states is that after the sample was used for diagnostic purposes, that they can use the same sample or the leftover material for research purposes. Unless you say, okay, I don't want this, and then you have to write to the director of the hospital. So that's basically these things make it in a way easier for researchers or for doctors to obtain these materials. But of course, it's kind of a struggle also compared to, well, and especially related to the use of the clinical data, because you can say, okay, the first patient, he is dead, but this one, he just came to the hospital, and okay, he had the opportunity to say no, but to what extent is that actively giving you the permission for the use of these samples, and also for the use of the data that are linked to these samples. And then I come to another important topic, and that's of course the data. The value of clinical samples lies in the data. Most researchers, you can have blood, But if the blood is not coupled to a specific disease type, if it's not coupled to whether the patient was a smoker or a non-smoker, it's often not possible for you as a researcher to address the questions that you're looking for. And you have multiple types of clinical data, and I just listed a couple, and you have all the data generated in the diagnostic lab of the or coming from the electronic patient file. What you also have is the personal data. And if I say personal data, then you say, anybody? It's a 4-letter word. Begins with a G. Indeed, the monster of GDPR. So basically, people are really afraid of the GDPR because you need a legal basis for processing all the data. So that kind of, well, people have the impression that that really shifts the way they can handle data. And in most cases, that's actually not really true. There were privacy laws in most countries, and in most countries, they were almost as strict as the GDPR is now. So for example, well, first this perhaps. I've been talking about informed consent. If people think GDPR, they think consent. That's the first thing that comes to mind, those little check boxes that you have to tick on your phone every 20 seconds nowadays. So informed consent is basically protecting ethics. So that's an ethical document that helps the ethical committee to revise, to make sure that whatever you're doing is in line with the ethical and legal principles. Consent, as it's defined in GDPR, is protecting privacy. So you have to, it's 2 times consent, but people tend to mix those, and people or researchers like to think that, okay, I've put 2 sentences in my informed consent form that I can use the data for research, that is sufficient, and that also giving consent for the GDPR related issues. But that's often not the case. Well, it can be for the primary research purpose, but then if you shift your research, you didn't ask permission for that specific research, or if you come up with a secondary line, you cannot use the sample for that secondary line unless you actively ask consent from that patient again. So in that sense, that would be very difficult to work with, and that's why consent, as it's defined by the GDPR, and the definition is labeled here, have freely given specific, informed, and unambiguous indication of the data subject to wishes, by which he or she, by statements or by clear affirmative action, signifies agreement of the processing of personal data relating to him or her. So this is basically consent. This is the checkbox thing. But of course, freely given. So for example, clinical trials, you're there in a hospital, you have cancer, your only treatment is this new drug that is now in this clinical trial. To what extent can you still freely give your information? Can you still freely give permission for the processing of the data? Answer is you cannot. There's pressure from the situation. So consent as a legal basis to perform trials or to perform research is often not the best legal basis. And there's 5 others, and if you read those 5 sentences, you see, for example, you need the data, or you need to process the data to save somebody's life. And for example, for a clinical trial, that is an acceptable reason. So there's a better legal basis than consent, especially for research, and that also allow us or give us more flexibility towards secondary use of the data. So for example, you shift your research line a bit, but you still want to use the same samples, you can probably find another legal basis that gives you the right to use the samples also for that purpose. And for example, there's the GDPR that allows the use of data outside the original scope if they pass a certain compatibility test. So if you explained the type of research to the patient, and the new line of research is really in line with the old one, then you can say, okay, it's compatible enough for the use of the data, or to be able to process the data also for that new research purpose. And we used that recently in Belgium for the presumed consent for the diagnostic route. So that little information leaflet that they got in the beginning of the hospital, for example, was one of the things that was thought, okay, this gives enough information to the patient, and together with national legislation, told us, okay, it's acceptable to still use this route. There's also exceptions for research. So research is often not considered incompatible with the primary goal. So although the primary goal might be very, very different, a secondary use for research purposes is usually acceptable and can be defended, especially if you link that to one of the other legal bases. So that means altogether that this active consent, this checking the box for GDPR is probably, in most cases, not a requirement, and that the use of material, the coupling of the personal data and the processing of the personal data coupled to this material is probably, in most cases, still acceptable also under GDPR. I'm stuck. Yep, that's the next one. Yep, thanks. No, not yet.
Maybe have disconnected. Maybe look at the slide, but this is still on.
Should be green. Yeah, I'll say when. I'm on this slide. That's okay. So okay, after the GDPR thing. So I already briefly touched on the importance of the data. So basically, you want all these patient identifiers or the clinical data of your patient to be able to answer specific research questions that you have in your research work. But then we have another concept, because you want information from the patient, but what is not acceptable is that you know actually who the patient is. And then we come to this concept of, well, it's basically, it's a two-way street. It's traceability. So you want to be able to trace back to the patient, but you also want to somehow make the patient, well, anonymous or pseudonymous. So if you look at this guy, as a researcher, you no longer know who this person is, so he's de-identified for you. But you can still use a lot of the information or all of the relevant information for your specific research. Then, yeah. So quickly about the difference between pseudonymization and anonymization, and both are defined by the GDPR. And basically, the pseudonymization is you put a key on the subject. So you de-identify, you make a code that de-identifies the patient for the researcher. And you basically make a list of the codes and link it to the names, and that list is put in a secure location. So the link is still there, but cannot be seen by the researcher. And that's the difference between pseudonymization and anonymization, because there you really break the link, so you can no longer go back to the patient. And then if you, that's key, press again. You can no longer go back to the patient, but actually the process of anonymization itself is really difficult nowadays, because you shouldn't be able, you have to remove all of the data that are linked to the patient that enable you to re-identify the patient. And for example, if you have this patient with a very rare disease, where there's only 4 in the world, and you know which hospital that person was treated in, you probably know which person it is. So related to research, anonymization is probably more and more becoming a dead end, and pseudonymization is probably the way forward. And especially in clinical trials, it's already embedded in good clinical practice. So in that case, it's already obligatory. And in clinical trials, anonymization is difficult anyhow, since its impact on the scientific usability of the data. In Belgium, and that's not always the case in other countries, but pseudonymization, according to the Biobank legislation, is obliged. We can no longer anonymize the samples unless with an explicit permission of the patient, and then together with the physician manager. And as an alternative, we can still go to the ethics committee and they can say, okay, it's acceptable. Oh, sorry. one back if you want. But then practically, and we see a lot of people struggle with this still. Who's responsible for the description key? So I don't know who of you is using human material at the moment. Only you? So none of the students? So who actually, talking to you now, so who's keeping the decryption key?
It's me, I guess.
Yeah. So normally it's
not anyone else in the room has access to this.
okay. So that's how it should be. So he's the best student in the class, of course. But we often see is that, okay, it's normally the principal investigator that is responsible for this key, but that the key is actually handed over to the PhD students, and that's actually also the PhD students that are going to look into the medical files of the patients, which normally legally is not an acceptable way of working. And also, well, and that's probably the same here, the practical organization is really on the level of the individual labs. So it's not that there's like an institutional filing system where these keys can be kept safe. Yeah, that really puts some safety issues on the availability of these data. Sometimes it's just a file somewhere in the well, either on a non-secured computer, or it's even lingering around, or is somewhere stored. But then the PhD student leaves and nobody still knows where that paper is. Or so these are things that we're still facing, and I guess not only in Belgium. So these are things that, if not taken care of in the labs that you're working in, is probably something that you want to look into and bring that up to the principal investigator. I added this in, genetic data. I was talking about pseudonymization linked to genetic data. That's basically a no-go. The amount of information that can be derived from a genome sequence is already huge and is only increasing. And I borrowed this slide actually from a colleague that well, we shared a talk recently, And he was a genealogist, and he said, yeah, the people had this under the Christmas tree. It was a family of 4. And then at a certain moment, the mother said, yeah, perhaps this isn't the best idea. So people don't realize what data is attached to these things. But at the moment, it's still limited. But in the near future, or very near future, the amount of data that you can mine from this is potentially huge. And especially if you go with companies like this, that information is spread relatively freely, can be sold. They actually sell this information. And you have to be aware that given a DNA sequence or a genome, you only need 1 in 6 in the world to be able to pinpoint every individual. So you can go back a number of generations. So the amount of information that you give away, not only for yourself, but also for your aunties and your grandfather and whoever is in within the first 6 generations, it's actually huge. And people don't realize that, so the importance of data, so GDPR is in a way there to protect your privacy. And people give their consent sometimes, or there's another legal basis to process these data, but people don't always realize how freely they give away their data, and that's definitely a risk. No, yeah, sorry. One back if you want. So traceability, I told you it works in 2 directions. And then I come to this concept of incidental findings. So as a researcher, at a certain moment, you might find something that is of importance to the patient. And then if you go to the next one. So basically, what is that? So previously undiagnosed medical or psychiatric conditions that are discovered unintentionally. And we see that a lot with medical imaging. And from personal experience, my father-in-law had this incidental lymphoma. So he fell from his boat. He was working that. The boat was unfortunately out of the water. It dropped down 4 meters. They took a scan of his body, and apart from the 7 fractures, they also found this lung tumor. In a way, he was lucky at that point because they could remove the tumor. But that is an incidental finding of value to the patient, so should be communicated back to the patient. Now, in this case, it was still as part of the medical examination. But if you, as a researcher, find information that might be of relevance to the patient, of course, the first step is to really reach out to the principal investigator to discuss it. And then usually you go back to some medical doctor, might be the physician manager of the biobank, for example, might be the doctor that treated the patient as part of the clinical trial. So you trace back that route to the patient or to the treating physician so that he can communicate this incidental findings to the patient. And that's also the case with genetic testing. But there we have the concept of risk. So in some cases it's very easy. You have gene X and you have a very high chance of getting a specific disease. For example, if you have BRCA, the BRCA gene, you have a high probability of breast cancer. So the risk level is high. I think that's something that needs to be communicated to the patient. But of course, a lot of these diseases are multifactorial, where you have like a 30% chance or increased risk of having specific disease, or and then it becomes questionable to what extent do you want to bring that information back to the patient, and also, of course, to what extent does a patient want to know? Because if you have a very high chance, then the medical need, the medical risk is high enough to say, okay, we have to do this. If it's only a limited amount of chance, then you might worry your patient. And so it's it's a difficult situation, and for example, in the Belgian biobank legislations, we have a number of times where the patient and the doctor overrule each other. The patient can say, no, I don't want to know, and then the physician can still say, okay, it's that urgent, you need to know. So this is something that is a difficult area at the moment. And the question is also, is it medically actionable? With the example earlier of the BRCA1 gene and breast cancer, we all know Angelina Jolie, and she removed or had her breasts removed. So that's in a way medically actionable. In a lot of cases, if you have a higher chance on a heart attack, okay, you can change your lifestyle a bit, but it's less obvious that that's medically actionable. Oh, sorry. And then that also comes back to the issue that the donor has the right to know, but then the question is, how do we bring that information to the patient? For the primary study, that's usually still relatively easy. You brought the initial information to the patient, but if you have secondary research lines building on that first line, how do you transfer that additional information to the patient? At the moment, I don't think people are doing that a lot. In clinical trials, the new regulation that, well, the aim is to make the clinical trials open source also to the patients, with the exception of some data, for example, the personal data. So there is some kind of portal, but for the academic studies, in most cases, those data are just part of the research and are not brought back to the patient. And that's also not that easy. Clinical trial-wise, it's the European, it's the EMA and medical agency that brings it to the patient as a whole. If each institution has to organize that on an institution level, that's probably too costly to develop. But that's another challenge for the future. And that brings me to the end and perhaps some conclusions. So there's a lot of sources of samples. If you have a research plan, you probably find the material. The GDPR is probably not stopping us or blocking us as much as we think it does. Pseudonymization is key. Forget anonymization, it will not work in the future. And of course, the link to the donors. I think if these are the 4 key messages that I provided you today, I'm happy. Thank you. Any burning questions?
So thank you very much, Peter. So it's time for questions. If you have some questions, now it's time for do it. So no questions? Julian.
Nice presentation. I would like to ask, for example, how is the case in fertility treatments where the donors receive, I don't know, an economic compensation for giving her egg cells? In that case, how it works with private companies and all that?
Yeah, that's a good question. That's actually a good question. I think about it before. We have we basically have a company in Belgium that does exactly that. So they store material as a commercial entity, and they indeed reimburse the people that provide the material. But I have to admit, I'm not sure how that fits with the national legislation at the moment. And the difficulty lies at the moment in the fact that the Belgian legislation changed recently, about 1 year ago. and became a lot more strict. And I don't think that's still acceptable in Belgium.
Working? Yeah. So my question is, when for a secondary use, that you go to a biobank and to get some samples from a patient, who is the owner of that samples? Is the biobank? Is the patient? Who is the real owner?
Well, depends mainly on the agreements. It's usually no longer the patient, although people like to think that. And sometimes we still say, okay, the sample is in a way owned by the patient, and if they would ask to destroy the sample, you still should. So in that case, it is owned by the patient, but it's not that the patient has any say about, well, they had a say about the use of the sample, but it's not that you have to go back to the patient each time for secondary use of the sample. So in a way, you have the agreement with the patient, and in that case, the ownership depends a bit on the arrangements that you have between the investigator that obtained the sample from the patient and, for example, the biobank. And what we, as a Biobank Antwerpen, what we say, for example, is, okay, we don't own the sample, we store this, we archive the sample and the data for the principal investigator, but they can also not themselves hand out the sample without permission from the biobank. So it's a two-way street, it's collaboration in a way between the two, and the agreements are really made between the biobank and the principal investigators on what conditions samples can be handed out to other research, for example, or even for their own research.
So because then if I need some samples and then some of them, I mean, I don't use it, what I have to do with that samples? I have to return to the biobank or I have to destroy it? Probably depends of the value of the samples, no? Mainly?
True. Well, you have the value of the sample, but also the processing steps. So what we as a biobank often do is, when the sample comes in, we divide it in multiple aliquots or in multiple slices of the same material, and we store those separately, and we only hand out the amount of material that is really required for that specific research. So the route that the sample directly goes to the researchers, that he processes the sample, stores it in a local -80 freezer, and then is able to reuse it time and time again. That is something that we want to get rid of actually in Belgium. It's also part of a new legislation. So in that regard, the sample quality is more guaranteed, and we say, okay, whatever we hand out to the researchers, the aliquots, let's say he asks two aliquots of material, since we cannot guarantee the quality of the sample as a biobank, we ask them to destroy that part of the sample, but then we still have the other aliquots for future research.
And my last question, I don't know if someone else has questions. My last question is, where's the limit in the agreement between the hospital and the patient about data? I mean, imagine that a patient has a cancer, and meanwhile we are doing research with these samples. No, the patient dies. Is his family or her family the responsible of that samples, or you're free to do whatever you want with this data?
Well, perhaps first of all, GDPR does not apply to people that are dead. So once you're deceased, GDPR no longer applies. So that's already a big big part of the story. So related to your personal data, you no longer have the option yourself, of course, to withdraw that information. And I think in most cases, if the family would reach out to researchers, he or she would still retract the sample from or the data from the system. Although we also have to be aware that in a way the data that have been generated by the researchers using that sample are in a way, for example, well, these are more owned by the researcher than by the patients. It's not his personal data anymore. So that data can still be used normally. So I think, yeah, the personal data would probably still be retracted if you really request that. But that normally shouldn't impact on the research results that have been generated already. So as soon as you've generated the results, then you can claim, also under GDPR, you can claim other legal basis for still being able to use those data as part of the research project.
So if there's no more questions, thank you very much, Peter.
Peter, very nice presentation.